reminders. So I'll let you know when you have 1 minute left and when to wrap up. So if you want to see those reminders, make sure you have your chat box open or in a place where you can see it. Also, if you're presenting and you have multiple slides in your presentation, please mention when you'd like me to advance to the next slide, because I'll be sharing my screen with your presentation. That way I'll know how to move through your presentation so it matches what you're saying. Also, in case you're not familiar with Zoom, there are some options with the view, so you can play around with what you can see in your screen. That should be, I think at the top of your screen, there should be options. You can like split it so you can see the screen share really big and then the people on the side smaller. So that's not super important, but something you can play around if you'd like to. As far as questions go, we'll have each presentation will be 3 minutes long, and then we'll follow that with 5 minutes of questions. Judges will be able to ask questions first, and then if there's time after that, we can allow audience questions as well. Audience members, you unfortunately will not be able to ask verbal questions, but you can chat them into the chat box, which should be at the bottom of your screen on Zoom. And if we have time, we'll get to those, and if we have time at the end, we can also circle back and address those audience questions. Let's see what else here. I think that's pretty much all for our reminders. Before we get started officially, would any of our judges like to just really briefly say their name and maybe what they do? You can go in any particular order.
Hi, this is Susanna Smith. I'm the Managing Director at the Institute for Advanced Study.
Thank you.
Hi, I'm Beth Kautz. I'm not sure how to get my picture showing.
There you go.
There I am. Okay, hi. I'm Beth Kautz, and I am in CLA in the Department of German, Nordic, Slavic, and Dutch. And I'm teaching a course on sustainability in Germany. And it was supposed to go to Germany in May, but we're not able to. So hopefully next year.
Oh, that's a bummer, yeah. Do we have any other judges in the room right now?
Pete?
All right, maybe we don't. Anna, how are we good to go ahead, or should we wait a few more minutes?
Pete is here. I don't know if he can hear us or not, but he is promoted as a panelist and he's judging.
Can you guys hear this?
Yep.
Okay, I'm Pete Swenson. I'm a U of M alumni and Director of Sustainable Enterprise at Tennant Company.
Awesome, thank you. All right, I think that are all the judges we have with us right now. So I think we can go ahead and get into presentations if that sounds good to you, Beth and Anna.
Yeah, hey, this is Beth Mercer-Taylor, and I'm excited to see the first ever online poster presentations and creative works.
Yeah, very exciting. All right. Judges and presenters, you can feel free to keep yourselves muted and your video off if you'd like during the presentations. And then when you want to speak after for the questions, definitely turn your microphones on and you can turn your video on too if you'd like to. I'll be sharing my screen so we can see the posters. And first up is, also I'm really sorry if I mispronounce names because I'm really good at doing that. So Aduramo Lasode is our first presenter. Are you ready to go?
Yes.
Yes, okay, thank you. I'll get your presentation queued up and then you can go whenever you're ready.
Sounds good.
All right, whenever you're ready.
Okay. So to start my presentation, I will give an overview of the motivation for this study, some objectives, the highlights of the methods used, results that came out of the study, and implications for sustainability. So the motivation for this study was previous research on survey of use of energy recovery technology in waste treatment. And this is focusing on anaerobic waste treatment because from the process of using microorganisms to treat either solid waste or wastewater, we can get biogas that has methane in it, and that could be burned or combusted to get energy or heat for further uses. Now, my research group collaborated with civil engineering here at the University of Minnesota, and they developed a novel process that generates methane and hydrogen. And so the question was, how do we use these or leverage it in energy recovery? So that was the previous study that was done. And you see the outcome of the technology curves for each energy recovery technology that is used for this process in the first figure on the upper left. Now, that also informs the regime curve, which shows more of commercial availability and application in terms of electric potential, electric power output based on wastewater volume. So those are the two outcomes of that previous study. Now, moving on to objectives. This study focuses on 3 types of waste sites. So wastewater plants, landfill sites, and dairy farms. And the idea is most of these places just flare their gases, and methane is a greenhouse gas, and that is dangerous for the environment. And so we're trying to evaluate the power potential for using these technologies in these sites. So the method used is a decision tree from the previous study that maps out how to use appropriate technology for each waste source. And the results show that for wastewater from domestic and industrial streams, domestic streams have lower energy potential, industrial streams have higher potential for using energy recovery. Landfill sites have way higher power on the gigawatt scale. Dairy farms are really prime for the use of these technologies because they generate a lot of methane. Now, moving on to implications for sustainability. One is there's value in adapting more waste to energy technology based on these results. The second is we need to reduce greenhouse gas emissions by using this technology. And the third is there is need for development of technology because within this systems, you have some individual sites that become smaller. And so we need to develop more technology towards the left of the regime curve shown in the motivation. That's an introduction. So I would open up for questions.
Thank you. Judges, feel free to unmute yourselves and ask any questions you have.
This is Pete Swenson. I think it's a great piece of work. And I wondered if you and Dr. Northrup and the company that you worked with are aware of this Centerpoint Energy program that they're kicking off now.
I think that the area tech person have been discussing their relationship with Expo rather than Centerpoint, but I can look into that. If you want to send me information, I would gladly take a look as well.
I'll send it to Jessica and she can pass it along to you. They're kicking off a pretty significant program, and they have a pretty broad spectrum of customers that maps on Excel's territory for electricity, but also is broader than that too.
I can't actually see very clearly on my screen a lot of the material in the poster, which is really too bad because it looks lovely. Could you explain in a little more detail what exactly is being demonstrated in the results panel?
In the result, based on the amount of biogas generated from these systems. So for example, the amount of methane that, for example, a landfill generates. What is the power output that we can get from an engine that uses that methane gas? Is power that you can get out once you run one of these recovery engines, and the input is the amount of gas from the site. Does that answer your question?
Not quite. Could you just tell me actually what the X and Y axes are? Because I can't read any. I can see the dotted lines going up, which looks like a positive thing, but I'm just curious what the axes are. You might have said that, but you were cutting in and out when you answered.
Oh, sorry about that. So the x-axis is amount of gas in from the site. So methane or hydrogen, the amount of gas from the site. The y-axis is the power, so like in kilowatts, that you can get from using that gas. And each dot is a site data point.
Okay, thanks.
Mm-hmm.
Hi, this is Beth Coates. This is all really kind of new to me, and so I'm trying to wrap my head around, did you describe what the technology or the engine looks like, or is that part of your presentation, or just the results?
So the technology was described in a previous study.
Okay.
So they're typically turbines, gas turbines, internal combustion engines, fuel cells, and micro turbines. So those are the ones highlighted in the previous study, because a survey of commercially available technologies was done to map the decision that you see, this decision tree you see in the methods. And so that is applied to the individual sites to say, what technology can we use at these sites? And based on the large scale for landfills, for example, steam turbines and gas turbines would be more appropriate. So that's the idea. And that's highlighted in the conclusions as well.
Okay, thank you.
Yep.
All right, thank you. I think now we'll move on to our next presentation from Amy Wesolowski. And this is a pre-recorded presentation, but Amy, I think, is here to answer questions afterwards. So I'll get that queued up right now.
Hello, my name is Amy Wesolowski. I'm a sophomore in the College of Science and Engineering, and the title of my research project is Co-Selection of Metal and Antibiotic Resistance Following Nanoparticle Exposure. My project is surrounding nanoparticles and their potential environmental effects, specifically whether or not they could induce resistance in bacteria, which are at the lowest trophic level of the environment and affect a lot of different environmental processes and just general health of the environment. So some background about nanoparticles: they're materials that are between 1 and 100 nanometers, which means they have a very large surface area to volume ratio. This said, they're very useful in a lot of different industries. They're used as catalysts in a lot of different experiments, which means they're also found in batteries because they transfer energy very well. So the problem that I'm looking at is, as I mentioned before, whether or not nanoparticles can induce resistance in bacteria. So a study that I read about was using Shmonella oneidensis and a lithiated nickel manganese cobalt oxide, or NMC, nanoparticle found in hybrid car batteries. The nanoparticle was toxic to the bacteria at a concentration of 5 milligrams per liter. So I'm using the Shmonella oneidensis bacteria because it can survive in both aerobic and anaerobic conditions, and it's found all around the world. So it's a good reflection of a variety of diverse environments and recycles minerals in soil. So the goal of my project is to find the minimum inhibitory concentration, or MIC, of a panel of antibiotics that have varied mechanisms of action, or ways that they affect a bacteria in order to hinder its growth, which would show me by what mechanism the bacteria actually become nanoparticle resistant. And I'm doing this using my array of antibiotics. Specifically for this project, I use 5 different antibiotics: erythromycin, kanamycin, metronidazole, novomycin, and puramycin. So in terms of results, I received varying results. So first, erythromycin showed to be successful. The control bacteria had an MIC of 3 to 4 milligrams per liter, whereas the NMC resistant had a concentration of 30 to 36 milligrams per liter. The same thing goes with kanamycin. The control had an MIC of 8 to 10, whereas NMC had an MIC of 160 to 180 milligrams per liter. Metronidazole showed similar results, but however, the NMC grew at all concentrations, which showed the possibility of contamination on the plates or in the cultures. But the control had an MIC of 60 to 80 milligrams per liter. Novomycin showed the same results for control and NMC in terms of MIC, so those proved to be somewhat inconclusive. And then puramycin, lastly, showed no signs of MIC whatsoever, which means that either the antibiotic was ineffective or simply that it didn't work with our specific bacteria. So analyzing this data, the results showed that erythromycin and kanamycin had the best results in terms of my hypothesis that NMC would grow at higher concentrations of antibiotic, and the specific mechanisms of action of those two respectively are protein synthesis and DNA synthesis. So therefore I can assume that the mechanism of nanoparticle resistance has something to do with the creations of protein or DNA. The NMC strains were able to grow at higher concentrations, all different types of antibiotic. So in conclusion, I found that nanoparticles would not be a suitable replacement for antibiotics, as they do cause resistance in bacteria. A possible extension of this would be to explore the structure of Shmonella oneidensis itself, to see processes that it undergoes for metabolism could coincide with resistance. I also noticed that the bacteria actually filament or elongate after exposure to the nanoparticles, so that could be interesting to look at as well. And I also believe that it would be beneficial to perform tests on a wider array of antibiotics with different mechanisms of action. So overall, I found that nanoparticles are noted to induce resistance in bacteria, specifically Shmonella oneidensis, a gram-negative bacteria, and therefore they can disrupt different aspects of the environment, potentially harm immune systems and health of animals and plants, and even bioaccumulate in the environment. So I believe that it's very important to look at new materials that are being created and analyze their potential long-term effects in order to protect the environment, humans, and general health of the population. And so I would like to especially thank the Center for Sustainable Nanotechnology and the Undergraduate Research Opportunities Program of the University of Minnesota just for letting the Carlson Lab continue our exploration of what nanoparticles do and what their effects are in our world. Thank you.
All right, if we have any questions, feel free to go ahead and ask those now. Also, if the presenters would like to turn their videos on while they present, they're more than welcome to do so.
Hi, by the way, this is Amy. Sorry if you couldn't see any of the boxes. They are kind of small on the screen, at least for me on my end. But if you have any questions about the results or what it says in any of the different sections, you're more than welcome to ask.
Amy, this is Beth Kautz. I'd like to step back and take a wide-angle view of what you're doing. Can you help me understand where do I find, I guess I'm just trying to figure out in what industry or who does this affect in my daily life? Where might I come across the things that you're talking about?
Yeah, for sure. Well, I have a little chart in the introduction section. You can't really see it, but it kind of describes the different industries that nanoparticles play a role in. As I mentioned in the beginning of the video, they're very prominent in catalysis and work very well for energy transfer. So that's why I looked at the NMC nanoparticle, which is found in hybrid car batteries. So those nanoparticles aren't found in large quantities in the environment right now, but in the future, once we start converting more to electric cars, those could be released into the environment in large quantities and then could affect a lot of different life in especially waterways because depending on where factories that make these batteries or nanoparticles are located, those could go into our waterways and affect animal life there, causing resistance and toxic effects because nanoparticles are built from different metals and ions that are kind of like layered together to form this nanoparticle. Other than that, nanoparticles are also used in a lot of clothing because they have antimicrobial effects. So that's why they're kind of linked with antibiotics in that sense. They both do a similar thing in limiting the growth of bacteria. And they're used in, a lot of different technology industries because they're so small, and healthcare industries are looking to their use as well for different medicines and drugs.
Thank you. That helps a lot.
Hi, Amy.
Hi.
How did you guys decide which sample of antibiotics to use in the study?
Yeah, so for narrowing down which antibiotics we would use, I first created a library of all of the antibiotics that we had in our lab, and then some different ones that had varying mechanisms of action or ways that they cause the resistance in the nanoparticle, ways that they attack a bacteria in order to kind of kill them off. And I looked at the Shigella onidensis bacteria. It's gram negative, so that affects what I was looking for in an antibiotic because the antibiotic has to be effective against gram negative bacteria. And then I inputted data into the library for each antibiotic and compared solubility, mechanism of action, whether or not it works against gram negative. And then I think there was one other category that we used to determine that. And then we ordered 5 of the 5 different antibiotics with varied mechanisms of action that were soluble in what we wanted to dissolve them in order to plate them onto the agar plates. And from there, we did test a few more preliminarily, but once we ruled those out, we just moved on to the 5 that works best.
Thank you.
All right, we're going to move on to the next presentation. If judges have more questions, we can certainly connect you with the presenters afterwards for a little bit of follow-up. But thank you, Amy. Next, we're going to hear from Chandra and Jennifer. I'll get your presentation ready to go. All right, whenever you're ready.
Okay, so our project started in the fall when we were accepted in the Institute on the Environment's undergraduate leaders program. Our project was originally focused on creating more green spaces to alleviate the effects of the urban heat island. for low to moderate income neighborhoods. But then we learned about the CREATE Initiative's research at the Humphrey School of Public Affairs on green gentrification and knew it was only appropriate to switch our focus. We decided to focus on instead capturing green gentrification in Rondo and Frogtown. I chose this place because I've lived in the Twin Cities my whole life and am a frequent visitor to the area and the local businesses. I also enjoy introducing friends and family to the area and bonding over the wide variety of foods and the urban green spaces installed by local community organizations.
So when we decided to work on green gentrification, we knew that we would have to partner with a community organization to learn more about the area. So we decided to partner with the Urban Farm and Garden Alliance, which is a community organization that is based in Rondo. So we work closely with Melvin and Mary in that organization. And through this, we were able to gain access to some of these locations that you see in the photographs. So we chose these locations based on green gentrification, if you couldn't read the poster, is kind of this idea that a lot of green development or sustainable development is viewed as apolitical, but is often used to push long-term residents out of neighborhoods. So we decided to do discussion through photography of what green gentrification looks like. So areas that either don't contribute to green gentrification or do contribute to green gentrification. And each of these areas is in those neighborhoods. And the photos are in black and white just because green gentrification is normally viewed as green space, but it can also be development that has sustainable aspects but isn't necessarily green, but is justified through sustainability. And we decided to highlight that by putting the photos in black and white.
All right, are you ready for questions?
Yes, if you could also just keep scrolling through the photos. I think we have about maybe 4 more.
Sure.
But we are open to questions now.
Hi, Susanna here, and I'm sorry I was trying so desperately to open up all of your pictures on a different screen because my anyways, I got 3 computers going here, so it's kind of crazy. What were your conclusions? Could you say say a little more about the aims and what you've concluded from that?
Sure. I can talk about that. So one of our goals was just kind of we really struggled with what the difference between like green development that was for the community and green development that kind of pushed the community out, what the differences between those were. So we talked to an expert in one of the UMN's grad programs to kind of figure this out. And she said something that was really useful is that green space that is for the community kind of focuses heavily on what the community needs, which might not necessarily be like as fancy or as in-depth or like as commercially oriented as development that would not be for the community. So part of our project was kind of looking at spaces that might possibly be developed and discussing what could be developed that would be more community oriented.
I'd like to follow up on that. This picture, the screen that's up now, the last paragraph is really helpful, talking about this difference between green gentrification versus community-oriented green spaces. I'd like to ask about Did you go out and walk the streets and find these spots yourself, or to what degree were you involved in visiting perhaps community organizations, local neighborhood meetings, and getting direct input from the community? So to what degree was it purely observation, and to what degree were the people living in the community involved in your research?
Yes, so part of the goals of our project were to gather community stories for this and talk about how it affected the community, but we were unable to do so due to the fact that was going to be the latter half of our project due to the current COVID-19 situation. But we were able to interact closely with many community members through our work in the Urban Farm and Garden Alliance. So we attended weekly meetings there with community members and also got to know their other projects that they were working on, and we're planning on attending their annual peace celebration before that was unfortunately canceled due to the current situation as well.
I can also add on to this. With working with Urban Farm and Garden Alliance, they really helped us identify locations that were meaningful to the community, and we also spent 2 days just exploring the area for about 5 to 6 hours and taking photographs of areas that we felt either inspired areas that move away from green gentrification and benefit the community, or instead hurt the community. So one of my favorite ones is their project behind the pedestrian bridge. I see that's also one of the questions down below, but the idea behind that photo was basically to capture the idea of reconnecting Rondo. And right now the bridges are very narrow, so it's really not enough room for 2 people to walk across together. So the expansion of the Dale Street Bridge right now is to create a art form of reconnecting the community by adding in pedestrian and bike lanes with turn lanes as well, and then art installations in various languages that say "We are Rondo," and 2 sculptures of metal trees on the side. So again, a lot of our locations were inspired by listening to people that are ingrained with working within the community.
Yeah, and I'll just to add on to that. So the pedestrian bridge that we photographed is kind of an example of something that tried to be community oriented, but maybe wasn't successful because it didn't take into account the community's needs. For instance, a lot of people use those bridges to go to their religious gatherings, but people in wheelchairs, it's too narrow for them to go across. So while the Dale Street Bridge isn't developed yet, when they widen the bike lanes and widen the paths for pedestrians and add art that is from the community, that would be an example of development that is sustainable in nature, but is oriented towards the community.
All right, thank you both. We're going to move on to the next presentation, which is from Will. So Will, I'll get your presentation ready to go, and then you can take it away when you're ready.
Alrighty, hello everyone. My name is Will Macheel and welcome to my poster presentation. In the absence of federal action on climate change, states are leading in climate policy. One such policy is the use of the social cost of carbon in electricity regulation. And so my research paper has 2 goals: determine the political economy variables driving states to use this policy, then using those significant variables, rank states that have not yet adopted this policy by their propensity to follow this policy trend. This will help advocates of SCC, or social cost of carbon policy, focus their resources on states that are more open in the political economy sense to adopting this policy first, leading to a domino effect of policy adoption. And so for my method, to achieve my goals, I constructed a statistical model, specifically a Cox proportional hazards regression model, that analyzes how different variables affect how quickly a state adopts SEC policy and electricity regulation. So what is the social cost of carbon, or SCC? The social cost of carbon is a tool used to quantify the impacts of climate change, such as sea level rise, droughts, and other extreme weather events, and it's measured by dollars per metric ton of carbon dioxide. During the Obama administration, there was an interagency working group of economists, climate scientists, who came up with an estimate of around $42 per ton of carbon dioxide. And so governments can use this estimate in market-based climate policy to encourage emissions reductions as people face higher prices for carbon-emitting activities, leading to long-term climate sustainability for society. So how are states using this measure of climate damage in electricity policy? The first way is integrated resource planning. So governments use the SCC as a cost consideration when improving their utilities' electricity generation plans. So coal would be penalized the most, followed by less dirty fuels such as natural gas, and renewables would not be punished at all because they do not emit carbon in electricity generation. The next is resource compensation. So in markets where electricity utilities buy from other utilities, they must compensate these utilities for the benefit of their avoided emissions at the value of the SCC. And the last and most widely applicable is cost-benefit analysis. So when governments are analyzing their proposed regulations and policies, they can factor in the value of the SCC as a cost or benefit depending on the policy. So alongside the right side of the poster, you'll find the results of my statistical model, which show that liberal, wealthier governments with relatively low electricity prices tend to adopt this type of policy, and high industrial electricity prices make a state less likely to adopt, likely due to the short-term interests of industrial firms hoping to avoid profit losses and the long-term concerns of governments about industrial employment leaving their state due to higher electricity costs from this policy. And relatively high residential electricity prices hurt the ability of states with average to above average incomes to absorb price increases and adopt this policy. Now, this does not mean conservative, poor governments like West Virginia with relatively higher electricity prices won't adopt this policy. According to my model, they just won't be the first ones to do it. So the map in the center, along with the list of high-ranking states on the right side, shows states that have relatively high conditional hazards according to the model to follow this adoption event using all the variables in the model. And to finish, since the creation of my model, a high-ranking state, Virginia, has actually adopted the social cost of carbon in integrated resource planning, confirming the predictive power of my model. Thank you, and I'm happy to answer any questions you might have.
Hi, Will. This is Pete Swenson.
Hello.
The $42 that you chose as the SCC, tell me a little bit more about how you decided to pick that number, and also would your findings or takeaways be different if you used one of the other numbers that is more like 125 or 135?
Sure, yeah, so a little bit more background on, that's a great question, a little bit more background on why I chose this number. So the interagency working group, as I mentioned, under the Obama administration, they came up with this number among a set of other estimates. And so how the social cost of carbon works is you look at socioeconomic projections, so population growth, economic growth, and how that relates to fossil fuel emissions, and how that relates to greenhouse gases accumulating in the atmosphere. And then of course you get climate change from that. And then they project those numbers out over a few hundred years and say, let's emit 1 ton of carbon today. How many damages will that cost in the future? And then we discount that back to the present. So the important part of that is the discount rate. And a little bit more on that, that's basically saying we value damages in the future less than we do today. It's pretty common in economics to use discount rates in finance outside of just climate policy. And so this $42 value is at the 3% discount rate. That's considered the central estimate as it's the discount rate related to consumption. You might see higher discount rates of 7%. That's pretty high. And so what that does when you have a high discount rate, that really devalues the social cost of carbon almost down to $1 in some cases. And so the three estimates for the year 2020, at the 3% discount rate, it's $42. At the 5% discount rate, it's probably around $12. And then the 2.5% discount rate is actually around like $65. And so you might say, as a government, we think that climate change is going to have more catastrophic effects than discounting it at 5%. So let's discount it at 3% so we can really account for and ensure that we are avoiding any unnecessary climate damages in the future when we're creating sustainable electricity policy.
Thanks. That's a great answer. Just FYI, if you add up all the subsidies for fossil fuels and do the, allocate them to carbon emissions, it's like $10 or $12 a ton. So that just sort of puts it in perspective.
Yeah, of course. Thank you. That's a great question.
Any other judge questions? We do still have about 90 seconds left.
Yeah, so I just want to make sure I'm clear on this. The idea is that like the state at greatest hazard is the one that would be the most likely to say, oh, this is something we want to adopt, right? And so that's where say people that want to change policy should concentrate their work. That's what we're going for here.
Yeah, that's correct. So as you see on the map, states trending towards dark red in color, like Pennsylvania, Virginia, Oregon, are the ones, according to the model, that are at the highest hazard of adopting. So that's correct. And so it wouldn't really make sense to focus your resources on a state like Mississippi or West Virginia, who, because of their political economy variables, would be pretty resistant to this policy change. And so as more states adopt, the theory says that other states that have not yet adopted, we'd be more likely to follow this policy trend, sort of like a domino effect. And this is a suboptimal policy. I would say this is until we have a national level carbon tax, which is more optimal because you're covering all sectors of the economy. And you don't have carbon emissions leaking out from one state that enacted the policy to other states that have not yet enacted the policy.
Great, thanks.
Thank you.
I'm trying to think about further implications. Is this the kind of research that you would share with political action groups or with government leaders, or where would you take this research?
Sure, multiple avenues, I would say. Definitely advocacy groups, so environmental interest groups, ones that work with public utilities commissions. That's where a lot of this work is happening. And so bringing those to public utility commissions proceedings, saying that other states are doing this. Your state has a similar political economy makeup. So say I'm in Pennsylvania, I say Maryland has done this, or New York has done this successfully. We share some similar energy infrastructure and political economy variables, so you should adopt this. And then also, I think government leaders should look at this as well and understand that this is a feasible policy they can enact until we have national level climate policy that really confronts the challenge of climate change.
All right, thank you, Will. Our last presentation in this session is Sasireka, which I think is a pre-recorded presentation, but the presenter will be around to answer questions afterwards. So yeah, we'll get started here.
Hi, allow me to introduce myself. I'm Sasireka Rajendran. You can call me Sasi. I'm a visiting scholar from India to the Department of Food Science.
Oh, I'm sorry.
Aspergillus.
Here, I messed this up. We're just going to restart. My apologies. I was trying to make sure that I had the computer audio on, and I got in the way. All right. Sorry about that. Sorry, everyone. PowerPoint's being weird in my computer, so I'm just going to close it out and restart, and then we should be good to go. All right. Here we go.
Hi, allow me to introduce myself. I'm Sasireka Rajendran. You can call me Sasi. I'm a visiting scholar from India to the Department of Food Science and Nutrition. So I'm here to present my lightning talk on characterization of Aspergillus species in red chili pepper using Fourier transform near-infrared spectroscopy. Of all the pathogens and microbes present, the reason I have chosen to work on Aspergillus is that Aspergillus itself is very potent and it causes severe respiratory illness to human as well as other livestock. It also produces aflatoxin, which has been identified as the second most potent carcinogen in the world. If I have to compare the ill effects caused by Aspergillus at this current prevailing situation, I would say it would be similar to the effects caused by coronavirus. Yes, it weakens our immune system and it causes respiratory problems. But one good thing is, it is not easily spreadable as corona, but the Aspergillus spores can be carried away in the air and it could be inhaled into our respiratory systems in all mammals. Aspergillus is a serious problem in developing countries like India, where I come from, due to improper irrigation, drying and storage. Groundnuts, corn and chili are some common crops that could be infected with Aspergillus species. So we tried screening red chili, which is an important economic crop in India, and it was found to have two major Aspergillus species, which are Aspergillus flavus and Aspergillus niger. Commonly used method which they currently use to detect these pathogens is PCR technique, which uses a lot of chemicals and it involves a lot of extraction procedures, which is also time consuming. Being a food science researcher, I would recommend using this NIR spectroscopy to detect these Aspergillus pathogens in red chili pepper within few seconds without requiring any sample preparation. To conduct this study, red chili peppers were collected, they were sterilized and were inoculated with the Aspergillus flavus and Aspergillus niger. They were then tested in NIR spectroscopy and the spectrum of these samples were collected. One positive and good result is that from the spectrum collected, we could easily differentiate between the fresh red chili pepper and the red chili pepper which has been inoculated with different strains of Aspergillus. So we have taken 3 different species and strains of Aspergillus. As we could see, this is for the fresh chili spectrum, and this is for spectrum of Aspergillus flavus, and this is the spectrum for Aspergillus niger. So through this method, we could clearly say that we can differentiate and also enumerate different types of Aspergillus using FT-NIR without any sample preparations. This is the calibration curve for the model developed to count the number of Aspergillus cells in red chili pepper. So we get the better model if we have better R-square value. So for the model developed, it has 0.91 R-square, which is very good, which also means that the error was only 10%. So I strongly recommend that this NIR spectroscopy could replace all the other chemical analysis method, and we could just move towards the sustainability by using this optical non-destructive method of analysis of Aspergillus species. I'm happy to answer all your questions in detail at this point. Thank you all for listening.
Hi,
Hi,
I'm
this
Sasi.
is Hi,
I'm
Sissy.
open to questions now.
This is Beth
Hi.
Kautz. There's quite a convincing report there. So my question is, what is standing in the way of everybody getting this device and using it?
So this is kind of only being used in lab models, and it is not like AOAC approved technique to analyze and enumerate the pathogens so far, since it required a lot of improvisation. Very recently, people have started working with FT-NIR, and it is making a huge progress these days due to the faster results and accurate results. And we've initiated this method development to identify the Aspergillus species in Red Chili. Once it is done, I think we can use hand-operated devices to just enumerate and isolate the pathogens from the good quality lots, I feel. Is that, did that answer your question?
Well, I'm wondering, I mean, are there economic considerations?
Yes, but only the initial investment is higher compared to other setups. But yeah, the operating cost is very, very low. And we could really use this method. I would strongly recommend we can be going with this method to determine the Aspergillus or aflatoxin content in any agriculture and horticulture produce for sure.
Okay.
Sasi, two questions.
Yes.
Do they use spectroscopy for any other bacterial identification is the first one. And the second one is, what is the distribution mechanism for this one? Is it fecal carried? Is it a fecal borne?
The answer for the first question is yes, they do use the spectroscopy method for identifying all the fungal and bacterial infections. So Aspergillus is a kind of fungal infection, so we could definitely use this technique for the fungal identification. And the second question, it is not like a fecal thing. It is like spread from the soil, and with improper irrigation and fertilization, the stress and everything causes the growth of Aspergillus in the plant itself before harvest, which is being carried away post harvest. And we when we don't consider removing that from the lot, it affects the entire lot, and there would be aflatoxin production in the produces during the production stages and processing stages. So we recommend removing that infected samples from the good quality lot to avoid further contamination and to get better end product in the end.
Thank you.
Thank you.
Thank you.
Sasi, does the Aspergillus affect the way the peppers themselves look and taste? I mean, are they harmful for the peppers? I understand that they would be harmful for any the things that eat the peppers, right? But the peppers themselves, is the crop harmed by Aspergillus being in it?
So the one way how the Aspergillus is getting into the pepper is, what we do in India is we harvest them when they are green, and we usually sun dry them in the field, like roadsides, since it is a developing country and we don't have proper drying storage facilities. So when we dry the green chili peppers in the road with different moisture content and rain and different change in moisture content, we get a lot of growth of Aspergillus, and it is not directly affecting the plant, but it is only after the post harvest when we consider this red chili pepper.
Thanks.
Thank you.
All right, thank you, Sasi, and also all of our other presenters. Really quick, we'd like to take a group photo of the presenters if we could. So would you all be willing to turn your videos on and we'll take a screenshot? Thank you.
Okay, this is Beth. So hang on a second. We're gonna get ready to do this. Jess, can you tell me, do we have all the presenters?
I think we're waiting for Jennifer. There we go. It looks like we're good now. Yep.
It's not letting me start video because it says the host has stopped.
Oh. Is that better?
Let me try again. Let's see. No, I keep getting you cannot start your video because
we can see you.
You can?
Yeah.
Oh wow, I can't see myself.
Oh, maybe not.
Look at the camera.
Okay. I just took a test photo.
All right.
Try again though.
There I am.
Oops, that was.
I think we're.
Hang on. Oh, don't go away. Don't go away, everybody. We're going to try this again. Okay. That was good, but I'm going to do it even again. So.
Well, we need Anna.
Your favorite pet, everybody. That was great. That was a good question. Thanks for bearing with us. And we'll be posting this, but let me do one more here so everybody can keep their eyes open. All right. That was me taking all your photos. Jess, do we have other things to do or should we go to?
Yeah, we are technically in break right now. The next session starts in 4 minutes. So if you'd like to stick around for that, you're welcome to do so. Judges, I think those are all the presentations you need to judge, but you're welcome to stick around if you have time or you're interested. Thank you so much for joining us today.
Everyone, thank you. This was so awesome. You. Beth Mercer-Taylor from behind that pink photo. What an incredibly impressive group of presentations. It's the first time we've ever done posters online. And I know there were some glitches with seeing everything, but you presented so well. Wow. Thank you.
Thank you.
Yeah, thanks everyone. Really great work. Keep it up.
Thank you, presenters and students.
Enjoy break, and remember that there's a panel starting at 2:15 in room 1, so you can rejoin for that as well.
All right. Thanks.
Bye, everyone.
Bye.